Hello everyone, Brian here today, and in this video I will be explaining exercise 5-19, which is known as the triple sugar iron agar test. Now for this test you'll be working with 4 different organisms here, the first one being A. faecalis, the second being E. coli, and finally M. morgani and C. furundi. Now once you have all of your bacterial cultures with you, you will then need to get a tube rack alongside a inoculating needle, not loop, and your Bunsen burner. And once you have that, you will then need to grab 5 TSI agar slants. Alright, so once you have your material with you, you can then begin inoculating your slants. And I'm sure you guys are very familiar with this part here. First things first, you'll always want to inoculate or sterilize your needle. And then flame the tip before grabbing the culture itself. And another thing that I want to mention that is unique with this test is that we always tell you to be very careful with how much bacteria you transfer over. However, for the TSI, there is a 2-part inoculating step which is going to require you to take a little bit more sample than usual. And I will get into that. So just grab a decent amount of the culture. Once you have that, you want to flame the tip and cap off your bacterial slant. Of course, prepare the TSI. And the first thing we're going to do is stab the slant. So you want to place your needle centrally above the agar itself and just gently puncture the media. And go as far down close to the bottom as you can, but not exactly touching it. And once you get to that point, you can then gently pull the needle out, making sure not to damage the agar itself. Okay, so once you have completed stabbing the agar, you will then perform a fishtail streak, just going back and forth like that in a zigzag pattern until you get to the tip of the slant itself, where you are officially done inoculating the media. Now you will then, of course, want to sterilize your needle before performing any more inoculations with your slants, and then cap it off just like that. And once again, make sure not to cap it off completely, as you want to allow for some oxygen to flow through. And you'll repeat this procedure for the next 3 slants, of course, excluding this last one here, which will be your control. Alright, so my computer was a little bit slow right there, but we are now back on track. Okay, so hypothetically, if you were to come to lab, your test results should look something along the lines like this. Now there's a lot of colors here, and I understand the TSI can be a little bit intimidating. It was a very difficult test for me to kind of fully grasp and understand. But kind of thinking about it in retrospect, I realized it wasn't that difficult at all. Instead of trying to memorize all the potential results that you can get, I realized it's just easier to kind of ask these 2 simple questions when approaching the test, and everything just kind of follows alongside it. So those 2 questions are, 1, what kind of sugar metabolism do you see here? And that will be represented by the different colors that you can see, like this yellow here, or the pink that you see here. Finally, the second question you want to ask when observing your test results is whether or not sulfate reduction has occurred. And that you can tell by this black precipitate that forms here, which occurs from the interaction between the hydrogen sulfide that the bacteria produces alongside the ferrous sulfate, which plays the role of the indicator within the test media. And finally, these colors that you see here are made possible due to this phenyl red indicator, which turns yellow when the environment is acidic, and can also turn pink when the environment is basic. So I think the best way to kind of explain this test and the theory behind it is to kind of go over each individual test one by one. And I apologize, I forgot that this part was also included in this slide. Reversion is very important. What you're seeing here, where the top half of the slant is pink compared to the bottom half, which is yellow, is due to this process known as reversion, and that occurs because there is a very limited amount of glucose within the TSI medium compared to the lactose and the sucrose. Now what happens is this test is incubated for a total of 24 hours. However, because the amount of glucose is so limited, the bacteria is able to consume all that glucose within a 12-hour period. Now for the next 12 hours, bacteria that lack the ability to use lactose or sucrose will then utilize the animal proteins within the media itself. And now, as you know, when bacteria break down proteins, they produce a lot of basic byproducts, which then accumulate in the media and make the environment more alkaline. And as I mentioned earlier, when phenyl red is in an alkaline environment, it turns pink. So this whole slant used to be yellow due to glucose fermentation, making the environment very acidic. However, once that glucose runs out, the bacteria will then have to utilize a different energy source, and that is through protein, because it does not have the ability to kind of tap into this secondary and tertiary sugar source because it does not have the genes and the enzymes to do so. Alright, so why don't we look at these tests one by one, and hopefully through this video you'll get a better understanding of what's going on here and you can feel confident making your own results and interpretations. Now one thing I want to mention here is that this is a very simplified and kind of annotated version of what you would have to end up writing down in your observations and interpretations table. So please make sure to look at the lab manual to really see what needs to be written down in order for it to be a correct interpretation, as that is very important. But just for simplicity's sake, I've just kind of just made things very short just to kind of get the idea across. So alright, let's jump into the first one here. So in this test, what you see is that the slant is entirely yellow, both in the top half and the bottom half. And what that tells you is that the glucose, first, of course, the bacteria first used glucose, as that is the preferred source of energy for most bacteria. And after 12 hours, when the glucose was all used up, it was then able to activate certain operons to produce enzymes, allowing it to utilize the lactose and the sucrose within the media itself. Now, of course, during this whole period of time of 24 hours, it's just fermenting all of these sugars and just constantly producing these acidic byproducts, keeping the environment acidic until the time you pull the test out of the incubator. And that's why you see that compared to this one, where we talked about reversion, it has maintained that yellow color. And so what you would write down, of course, are these simple symbols here. And there are two, because the first symbol represents what you see in the top half of the slant, and the second symbol represents what you see in the bottom half. And because both the top and the bottom half are yellow, you would then write down A, which stands for acidic, and of course A for the bottom half as well. Alright, so for the second one here, we can see that both the top and the bottom half has turned pink. And this shows you that this bacteria is not able to, one, ferment the glucose, and of course lacks the ability to ferment either the lactose or the sucrose, and so has just kind of used the protein within the media itself, turning the entire slant pink. And in order to, and thus you would then write down K, which stands for alkaline. And because, as I mentioned earlier, both the top and the bottom is pink, you would then write K/K. Alright, so for the third tube, this is the reversion tube. We know that the top half is pink because the bacteria was not able to use lactose or sucrose. It was only able to use glucose, and then once the glucose ran out, it then immediately began to break down the animal protein, turning the top half pink. And that happens because there's just more demand in the top half of the slant. And because of that, it consumes the glucose faster compared to the bacteria in the bottom half of the slant. And once the glucose here is depleted, it then starts using the animal protein within the media sooner. And that is why you sort of see that the reversion kind of starts at the top before, after an extended period of time, kind of finally reaching into the bottom half of the slant itself. So because the top half is pink, we know that's basic, so we would then write K, and the bottom half is acidic, so we would then write A. Now before I go into the fourth one, I forgot to mention here, this little white region here, sometimes what you'll see is that the bacteria will produce a gas. And that is notable in a variety of different ways. Sometimes it's just a small crack or these fissures that appear in the agar, or sometimes there was enough gas production where it kind of shifts the entire media itself up, kind of separating it into two halves. So that's kind of why I mentioned earlier when you are inoculating these tests to be very careful, because if you do end up cracking the agar, when you come to this point, you won't be able to determine whether the bacteria was producing gas because you cannot tell if those cracks were made by you or the bacteria themselves. Alright, so this fourth one here, what you're seeing is this black precipitate kind of overwhelming the entire test media itself, kind of disallowing you from seeing what the color is down here. However, that is not a problem because in order for this hydrogen sulfide production to occur, the bacteria needs to be in an acidic environment. So knowing that, you know that fermentation must have occurred because the media environment itself would not be acidic if it weren't for those acidic byproducts that happen through glucose fermentation. Also, even though we cannot see the bottom half of the slant, we can sort of kind of see past the black precipitate in the top half. And based off of the color here, we know, of course, since it is pink, that the bacteria was not able to use lactose or sucrose, and the media has now started to go through the process of reversion, where maybe at one point it was yellow because it was using the glucose, but now has began using the animal protein within the media, then shifting the pH environment to be more basic, and of course the indicator then changing colors to follow suit. So the symbols and what we would write down in your table would be K to represent that the top half of the environment is alkaline, and the bottom half, even though we can't see the color, we know that this black precipitate cannot have occurred if the environment wasn't acidic. We would then write A. And finally, because there is hydrogen sulfide production due to this black color forming, we would then write that down as H2S. And finally, your last two being your control, we know that nothing has occurred in here. Okay, so this will then bring us to the end of our video. I've just kind of summarized three core concepts that I believe is important for you guys to really understand and memorize in order to be comfortable with this test. And the first one being, of course, is to know why this test is being used. And this test tests for the bacteria's ability to use either sucrose or lactose, and to see if they have the ability to reduce sulfate as well. A lot of enteric pathogens within the family Enterobacteriaceae tend to produce hydrogen sulfide and also lack the ability to utilize sucrose or lactose. So, if you were to end up seeing the right color profile and that black precipitate forming within one of your TSI slants, you can be fairly confident to know that you are on the right track of identifying a potential enteric pathogen. Now, the second concept that I want you guys to really take away is reversion. Reversion, once again, is the color change that you see in the top half of the slant. That is occurring because the bacteria first uses up all the glucose because there's only a limited amount within the media. And after that 10 to 12 hour point and all that glucose gets used up, because it cannot or does not have the ability to break down the sucrose and break down either sucrose or lactose to ferment them, it then shifts to using the protein within the media. And of course, the basic byproducts that are formed then create this alkaline environment and shifts the color from yellow, which it once was because it was, of course, acidic after glucose fermentation, but then turns pink because of the because of the basic byproducts that are formed through catabolizing protein. And finally, okay, there is a slight mistake here that was a little bit careless on my part, but hydrogen sulfide production only occurs, cross that out, not anaerobic, only occurs in an acidic environment. Okay, so ignore the anaerobic, just focus on the acidic. And lastly, gas production, that can be determined based on, little cracks or fissures that you see in the media. So please keep these three things in mind when approaching this test. And the last thing I want to talk about is, in this video, we only kind of went over 4 potential outcomes. There are much more than that. And so I think it's important to kind of practice learning about the other potential test results that you can get through the TSI in order to be fully comfortable with the test itself. And I believe that will be it for today's video. I hope you guys found this helpful. And after this, you guys will then be starting your unknown projects, which I can say for myself, it was a very fun project. And I know some of you may be a little bit intimidated with it, but you guys have performed all the tests that have been in your lab manual now, and just know that these are all tools that you can use to help you isolate your unknown and find the species that you were given. So thanks again for listening, and I